Right, hello everyone. Yes, so I'm Robert James. I'm from the Advanced Process Technologies group at the University of Manchester. And today I'm talking about some recent work we've done where we're doing auditory pathway modelling on SpiNNaker hardware. So we had an excellent overview of SpiNNaker earlier, so I don't need to go into too much detail. Essentially, I want to reiterate the fact that a SpiNNaker machine is just made up of many ARM microprocessor cores arranged in this toroid donut-shaped network. And the unique thing about this hardware is we can communicate from core to core using this multicast routing mechanism, which is essentially a one-to-many communication method, which lends itself very nicely to performing digital simulations of spiking neural networks, where we'll see this kind of connectivity. But what you can do is you can use this hardware for other applications that aren't necessarily for simulating spiky neural networks. So this brings me on to the ascending auditory pathway. So this is the early stages of the auditory pathway. So going from a sound entering into the outer ear, passing through the middle ear, where it will cause vibrations in the tympanic membrane, which then move these inner ear bones, ossicle bones, which then feed into the cochlea, which in your ear is spiraled up, but for the purpose of this diagram, we've unrolled it. You can see inside the cochlea that depending on the frequency of the sound stimulus that you're applying, it will resonate in a specific area along the length of the cochlea. So this is the basilar membrane along here, which vibrates. And then the motion that is caused in the basilar membrane at different locations is picked up by these little inner hair cells which line the entire cochlea. And there's approximately 3,000 of those in a human cochlea. And what they're essentially doing is picking up the movement on the basilar membrane and converting this into the spiking signals on the afferent auditory nerve fibres. And there's about 30,000 of these in each human cochlea. So what we've noticed with, looking into this biology is that there's a lot of parallel processing happening inherently. So we thought that we could actually reproduce this on SpiNNaker hardware. So essentially, we can have many models, some modelling the outer and middle ear, then others modelling each individual section of the cochlear basilar membrane, and then finally other ones modelling each of these inner hair cells. So that's what I'm going to show you on this slide. So we can see what SpiNNaker looks like under the hood. So we first have a sound passing in to this outer middle ear model. So there's only one of these on a SpiNNaker machine. But then what this model will do, it will actually communicate its produced output, which it does through digital filtering, to many cochlear channel models, which is running concurrently on the same machine. So this is using this multicast transmission method to send that same value to all of these. Each of these will be tuned to a specific frequency, so that will correspond to a particular portion of the basilar membrane, and then it will send its unique outputs to its designated inner hair cell and auditory nerve models. The reason I have, 5 of these in this diagram make up a single inner hair cell. The reason I have 5 is so we can process this in real time, so the computation isn't too intensive. And each of these will produce 2 auditory nerve fibre outputs. So if this works, we should see a little funky animation. So we can see the sound going into the outer middle ear, and then the relevant ones feeding into the cochlear channels, and then each of those outputs feeding into our inner hair cell models, finally producing the spiking auditory nerve fibre outputs. And then the idea is that we can then directly connect these outputs into spiking neural networks running on the same SpiNNaker hardware. So we have a massively parallel sensory input feeding into a massively parallel spiking network. So the idea is we can then bring up the scale of these kind of simulations to biologically similar numbers. And we can see down the bottom here, we're able to produce simulations of 30,000 auditory nerve fibres, and this is across approximately 18,000 cores. This is what the output looks like in the form of a raster plot. And we can see we've split this in terms of the high spontaneous rate fibres and the low spontaneous rate fibres that the inner hair cells produce. It's worth mentioning as well that the actual algorithms I'm using for each of these individual models aren't their existing ones that are already out there and have been tested against a lot of physiological data. If you come chat to me in the poster session, I can tell you more about exactly what those algorithms are. So just to summarise, SpiNNaker simulates the early stages of the auditory pathway, but we do so without incurring any of the processing performance penalties that you would get if you simulated this on conventional computers. So you can't really scale up to the human number of inner hair cells if you need it to do real-time processing or anything close to that. The hypothesis I've got for further research is the scale of this parallel sensory input actually influences or increases our task performance in certain auditory tasks. So yes, and also the fact that we have real-time sensory input processing then leads to the potential for robotic applications. But the downside is, because of the low clock rate of the ARM cores that we have on SpiNNaker, it means I'm limited to an input sampling frequency of around 22 kilohertz for real-time performance. Okay, thank you very much for your time. Cheers.